So, hello and welcome to our online lesson. And we'll be continuing learning about non Mendelian patterns of inheritance. So, I have some stuff already set up here that I'm going to be referring back to, but I'll have my little portable whiteboard here and I'll just minimize that. And we'll pick up where we left off last. So, we did do the blood typing lab and we looked for agglutination to occur to see if there was a positive test for a particular antigen to determine the person's blood type. So, just a short review to start blood typing. And so, blood type is determined by surface proteins called antigens. So, there's two major blood types where we can look at the ABO system and we can look at the RH factor. So, blood type is determined by multiple alleles and codominant alleles. And so, we see the A allele, the B allele, and the O allele. Now, as you see here in the picture, type A bloods has an A antigen that's a surface protein on the surface of the red blood cells. And then circulating through the body, we see that there are these Y-shaped green structures, and those are antibodies. So someone with type A blood has A antigens and B antibodies that are circulating through their bloodstream. Someone with type B blood has B surface proteins and anti-A antibodies. Someone with type AB blood has both antigens, but neither antibody. And someone with type O blood has no antigen, no surface protein, but both antibodies. Right, so we did that before we left. And another thing we have done is, if you know you are phenotypically blood type A, can we absolutely determine your genotype? And we know that you can't. You could be homozygous A or heterozygous A, but that's not completely possible to determine unless you're looking at that individual's parents or children or other members of the family. How about if you are phenotypically blood type O? So if you are blood type O, there's just one genotype that would determine that phenotype, and so that would be homozygous O allele. A heterozygous type A woman and a heterozygous type B man have children show a Punnett square. And so we did that, right? So if you are a heterozygous A woman, here is capital IA, recessive I. If you're heterozygous B, here's I, capital B, lowercase I. And then the Punnett square is here. Now what's interesting is that the genotypic and phenotypic ratios they're the same, and they're both 1 to 1 to 1 to 1. Now how do we get that? So here, the genotype IA, IB, then IB, lowercase I, then IA, lowercase I, then lowercase I. That would be the 1 to 1 to 1 to 1 to 1 genotypic ratio. And then for phenotypic ratio, we have blood type AB, blood type B, blood type A, and blood type O. So that would be the 1 to 1 to 1 to 1 phenotypic ratio. So one thing that we did talk about before we left as well was that the A and B alleles are codominant. So it means that when the A and the B are together, they both are expressed. So they're codominant. And we talked about this horse. If you guys remember, it was called a roan. And this horse that is a roan has both red hairs and white hairs. They're equally visible. They're both expressed. So, blood type, the horse, the roan. We also, if you recall, we talked about these checkered chickens. So they were also showing codominant patterns of inheritance. And that's in contrast to incomplete dominance, where you may have a red flower, a white flower, and then the offspring could be pink. So those are just additional pictures that I had here. But the RH factor, and last week you guys should have copied these into your notes, but I thought it was important that I explain this to you. But the RH is another surface protein. And you can see a pretty good picture here. I like this because it shows how the protein is on the surface of the red blood cell. So we learned in another unit about how the cell membrane is pretty complex. It's made up of the phospholipid bilayer, and then there's proteins and carbohydrates. So the A antigen is one, the B antigen is another. But here is a different antigen called the RH factor that's also found on the surface of red blood cells. So if you're RH positive, you have that. If you are RH negative, you do not have it. And in terms of genotypes, it's just a simple dominant recessive pattern of inheritance. So what does that mean? Well, what it means is, it means that all you need is the one dominant allele in order to express that. So if you are big R, or big R, little r, that means you would be positive. Or if you are recessive R, then you would be negative for that blood type. And RH positive tends to be more common than RH negative in the Caucasian population. And so there are other proteins and carbohydrates on the surface of red blood cells. But when we talk about blood type, we are talking about the ABO system and the RH factor. So now, just to give an example, we will be getting to pedigree soon, but just to give you an idea of how this is all inherited, right? We talked about our A, B, AB, O blood type, and just to show you how it could be inherited and how it's passed down. In my pedigree that I have here, the squares represent males and the circles represent females. So here, what it means is that we have a father, a mother, and we have 3 children. We have a father, a mother, 1 son, and 2 daughters. So let's say, that this father is O negative. And let's say that this father's O positive and this mother is A negative. So then what would this daughter be? Well, this daughter could be a variety of different genotypes, but let's say that this daughter is blood type A positive. So the question is, if the daughter's blood type A positive, can we know definitively what her genotype is based on the parents? And the answer here is yes. Because this person is blood type A, it could be homozygous or heterozygous, but it must be heterozygous because the father is type O blood and only has the recessive I allele to pass down. The mother is recessive. She has 2 recessive alleles because she has RH factor negative. So here's the mother's RH factor, and the mom can only pass down that recessive allele. But at the same time, this daughter has blood type positive. So there's the dominant allele. And so in this case, we can tell what genotype goes with a blood type A positive because we have enough information from the parents to then determine that. Okay? So then there's a couple of other pictures, and now, polygenic inheritance is a type of inheritance where there's multiple genes that control the expression of a trait. So now I have to make myself smaller. All right, so there we go. And back here. Okay, so when we look at polygenic inheritance, this is when there is a trait that is determined by more than one gene. So here, the example could be skin color, it could be height, it could be eye color. I have that here. This is actually an even better table. But at least for eye color, there could be two genes. So here's one gene, which is HERC2. And if you notice that for this one gene, there are two alleles. One, big B, is brown, and the other, little b, is blue. So if you're big B, that would lean you more towards being brown. If you're little b, that would lean you more towards being blue. That's one gene. But then another gene that's important for eye color is GEY. So you see how green is dominant over blue. So big G is green, and then little b is blue. So eye color is complex. Here you see two genes, and you see a brown and a blue allele for this gene, and you see a green and a blue allele for this gene. And there's more than one gene that determines eye color. There's more than one gene that also determines skin color as well. Now, we can explore how the expression of one gene may affect the way that another gene is expressed. So, oops, sorry. Okay, I needed this. So when we explore epistatic patterns of inheritance, we're looking at how the expression of one gene may affect how another is expressed. So what you see here are you see these mice that are crossed. So this first mouse is heterozygous for two traits. Now, what are those two traits? So those two traits are basically, one is for the actual color of the hair, and then the other gene is for whether or not that pigment actually gets produced and distributed. So now let me make my whiteboard bigger. And so back to this. All right. So here are chocolate labs, black labs, yellow labs, and they also follow the same epistatic pattern of inheritance as well. So again, two genes, one that actually determines what the color is, and the second gene that affects the pigment production of that first one. So you have this picture in your notes as well, but the black lab has to have the big B allele, and it may have a second big B allele, but then it could also have the recessive B. So anyway, so big B is for black, and then little b is for the brown allele. And then another very important gene is what codes for the production of that pigment. So we have here, I'm going along with the letters that are in your notes. So we have big E and then little e. So big E is for the pigment that's produced, and little e is for none. So, for example, if we want to give the genotype for a black lab, well, that black lab could be big B, or big B, little b, okay? But that black lab must have pigment produced. So, all right, so we took care of the genotype, big B, big B, little b. Little b doesn't code for black, it codes for brown. All right, but we need to make sure that pigment is produced. Okay, so pigment production correlates with the genotypes big E and big E, little e. Now, if we have a genotype like this, that would correlate with a black lab. This would still correlate with a black lab because the big B codes for the black color, and then the big E codes for the fact that the pigment is produced. Now, let's say that we get rid of this. Let's say that we get rid of this, and then we replace it with two recessive E's. Now, even though this lab is supposed to be black, now the pigment is no longer produced, which means that it's now a yellow lab, which means that now the genotype would be matching a yellow lab. Or, let's say instead of big B, lily, let's say we have this now. But it doesn't matter whether I have the genotype big B, little e, or little b, little e. Whether it's this or whether it's that, both of these are still a yellow lab because in both cases there's no pigment that is produced. That's the point and that's the idea. All right, so now the next. All right, so problems to solve. Okay, now let me move myself again. Okay, all right, here and back here. So, what are the possible genotypes at both genes of the black dog? So, what are possible genotypes? So we started giving possible genotypes for the black dog. And it's anything that would have to importantly have a big B allele and anything that would have a big E allele. So possible genotypes at both genes of the black dog, it would be maybe big B, big E, big E. It could be big B, little b, big E, big E. It could be big B, little b, big E, little e. It could be big B, thinking on the spot here, big B, big E, little e, and I believe I got all of them. It's possible I might be missing one. But yes, so those are all different genotypes for the black dog because again, it has a black allele and then it has the pigment production allele. And now for the yellow dog, what are possible genotypes for the yellow dog? Well, it doesn't matter. It doesn't matter what combination. I can write anything here. I can write any combination of letters I want, but as long as there's two recessive E's, there's no pigment produced, and therefore it's a yellow lab. Now the brown dog, okay, so the brown dog, this one is important. Because the black allele is dominant over the brown allele, that is why if it's brown, it would have to have two of the recessive B's. But it needs pigment to be produced anyway. So a brown dog could be little b, big E, or little b, big E, little e. And basically, this pattern of inheritance is similar to the dihybrid crosses that you've done. It's a different pattern. Now, Other non-Mendelian patterns of inheritance includes those that are affected by the environment. So external and internal conditions can affect genetic expression of genes, so whether the genes are active or inactive. So body temperature, carbon dioxide levels, sunlight, chemicals, food. The reason why the leaves change color in the fall is because they're losing chlorophyll. The chlorophyll is breaking down, and that's because of the surrounding temperatures. So we talk about environmental effects on gene expression, and there can be all these different factors in the environment that affect how genes are expressed. So the Himalayan rabbit that you see at the bottom here is this white rabbit that if some hair was shaved from its back and an ice pack were placed there, the hair would actually grow back black and melanin would actually be produced. Here's a Siamese cat, and you see in the ears, in the legs, in the tail, it's darker. Its extremities are darker because those temperatures are lower than 37 degrees Celsius. But the core of its body is paler, and that's because it's warmer there. Now, identical twins may be born with the same DNA, but they're exposed to different environmental conditions remarkably as they grow older, and that does have an effect on how their genes, even though it's the same genes, are expressed depending on what particular trait it is. Now, this is very interesting. So, our genome is all of our DNA. Our genome is all of our A's, our G's, and our C's, and our T's. An epigenome is something special. An epigenome has these chemical marker tags on the outside of the DNA. And there's a picture here of a mouse, but it'll be better because I'm actually going to take you to the website. But these mice are affected by their environment. So what do I mean by that? Well, the best way for me to explain is to actually show you the animation. So we're coming to understand about our epigenome more and more, and yikes. All right, so this here is called Lick Your Rats. So, okay. This should be working.
Choose among the options on the left to learn more about how mom's nurturing affects her pup's genes, their brains, and their behavior.
Okay, so the reason why I'm sharing this with you is because I feel like it's an effective tool in order to illustrate what an epigenome is. So what you see here is you see that there's two rat pups. You see that there's a rat pup that's highly nurtured and a rat pup that's low nurtured. And so the difference is that the high nurtured rat mom is devoting a lot of attention to her baby. And the other one is not devoting as much attention as her baby. So what do we mean by the epigenome? Well, let's take a look closer because this is what's happening in the environment, but let's take an even closer look. So here is the epigenome. The blue and yellow, the blue is the histone proteins, and the yellow is the chromatin. I'm sorry, the yellow is the DNA and the blue is the histone proteins. Together, collectively, they make the chromatin. So what happens is these green dots are these methyl groups. And in the high nurtured rat pup, the GR gene has been kind of like uncoiled. And in the low nurtured rat pup, this GR gene is still tightly coiled. Well, what does that mean? It means that RNA polymerase can't go in and read the gene. so the gene can't be expressed. So, in the rat mother that licks and takes care of her rats, the genome is still the same, either one, but this one has been opened up so it can be read. Now, what goes on in the brain cells? So,
Inside the brain sits a structure called the hippocampus. One job of the hippocampus is to coordinate the body's response to stress. In the hippocampus, cells produce a little or a lot of GR protein, depending on how much the pup was nurtured. During the fight or flight response, the hormone cortisol is released into the bloodstream. Cortisol binds to GR protein, causing the cell to send out calming signals. Rats who have a lot of GR protein relax quickly after stress. Rats with little GR protein remain stressed for much longer.
so it's really important that I show you that because we keep talking about this thing called the central dogma. And it's like, why did you learn about transcription and translation? It's like, why are proteins important? Why do we keep talking about this? The mother, like who was taking care and nurturing the rat pups, actually influenced the expression of the GR gene. And so, as that was changed, the GR gene in this case affected how much of the GR protein, this blue thing right here, was made. In pink here is something called cortisol, and that's the cortisol hormone. So it's a hormone that we produce when we are in stressful situations. So when there's more of the GR protein, more of that cortisol stress hormone can be broken down. And then, now here's the adult.
Nurturing affects more than the GR gene. As adults, high and low nurtured rats have expression differences in hundreds of genes. In the end, what you get is two rats that behave very differently. Calm or anxious behavior can be an advantage in different situations.
So that is showing you the whole relationship between environment and gene expression, the connection between DNA and the proteins, at least here, that are made in the brain. So I hope that was helpful. But if we take an even closer look at how everything relates, thinking about like the central dogma, this here is the GR gene. And again, blue is the histone proteins and yellow is the DNA. And then here is the mRNA and here's the protein. So this gene can be read because it's opened, and the mRNA can be transcribed, and the protein can be translated. So here's the cell with a lot of this green fluorescent protein. So now I'm going to take the dial and I'm going to close up my gene. And what you see here is that my cell is no longer lit up because there's no more protein, because there's less mRNA, because I can't transcribe it from the original gene. I'm sorry, this is actually the GFP gene instead of the GR gene. But anyway, the idea is the gene is not being read and not being expressed. Now, let's go in the other direction. And here, this gene for the green fluorescent protein that you're going to see in the cell, let's open it up. So I'm going to turn the dial in the other direction, and as you see, as the gene is opened, more of the mRNA is transcribed. As you saw, more of the protein is translated, and the cell is brighter. All right, so let us wrap things up and finish up with this last pattern of inheritance. So, there are some traits in humans that are not so easily observable, but in cats, these are really observable. So cats are a great example to use to teach this. Now, early on in embryonic development, if you're a female with two X chromosomes, one X chromosome randomly becomes inactive in each embryonic cell. So here is, here's the zygote. And then as it develops, there will be one chromosome that kind of condenses and becomes like a little tiny body, like its own little body. Call it a bar body. And it's random as to which of the X chromosomes becomes inactive. So if we look at a calico cat where there's some orange and black alleles, maybe randomly it's the orange that becomes inactive, maybe randomly it's the X that becomes inactive. But the female who would have two black X's, whichever one gets shut off, they're all black anyway, so that female would appear black. The red female, the case would still be the same. But for a female that has one red and one black allele, there's this random expression of that allele on the X chromosome. For males, the male just has one allele because they have one X chromosome. Of course, there's exceptions with Klinefelters, but that really is the exception, and that's not the typical case. So that concludes this lesson, and we are not doing page 6 of the notes on the lag operand, so we are just going to skip that. It's great to do this online lesson. All right, good seeing you guys. All right, bye.